Okay, at this point what we need to do is actually shift our focus from the very specific to the more broad with the nervous system. And to do that, we're actually going to start our study of the brain itself. So the first thing we're going to look at here are these structures, the blue structures that are on the screen. These are probably the first time you've seen something like this. These are what we refer to as the ventricles of the brain. And as you can see, they look very different depending on how you view them. So the first ones we want to show and point out here on the diagram are these. And they are paired on either side of this feature here, which is called the longitudinal fissure. This is what actually divides the brain into left and right hemispheres. So left side of the brain versus right side of the brain. These two ventricles are referred to as the lateral ventricles. The next one here is the one that's in the center, and that is referred to as the third ventricle. And the last one is down here, right behind the cerebellum. And you'll notice that it does continue on down into the spinal cord, and this is the fourth ventricle. So what exactly is a ventricle? Well, a ventricle is a chamber that is filled with cerebrospinal fluid. And we abbreviate this CSF just because it's a long word to write. So this is the first time we've really had a chance to talk about what cerebrospinal fluid is and what it does. It surrounds and bathes the tissues of the central nervous system. So as it does that, it carries out three essential functions. The first one is cushioning. The second one is support. So it actually pushes up on the brain and it makes the brain float in the cranium. And this is actually very important because otherwise these neurons that are on the underside of the brain here would actually get crushed under the weight of the brain on top of it pushing down. The last thing that cerebrospinal fluid does is transport. And it transports things such as nutrients, hormones, and waste. Now, just to give you a little idea here about our cerebrospinal fluid, we have about 150 milliliters of volume in the ventricles and the spinal or the central canal. Most of the cerebrospinal fluid is made in the third ventricle. It's not that the others don't make it, it's just that the third ventricle makes more for whatever reason. But as you can see, they are all connected, so they all assist in the circulation of that cerebrospinal fluid. Now that 150 milliliters of volume that we have is actually replaced. We replace it about every 8 hours. So in the course of a 24-hour day, we basically turn over our cerebrospinal fluid about 3 times. We make about 500 milliliters per day. Just to give you some numbers that kind of coincide with that. So those are our ventricles. So now let's start taking a look at some of the regions of the brain you're probably more familiar with. The first one that we want to look at is a sagittal section of the brain. And so basically what you've done here is you've actually cut down, including the skull, along the longitudinal fissure. And so you basically have divided, this is a longitudinal cut, or sagittal cut, you've actually divided the brain into a left half and a right half. So we're looking at the cut exposed side of one of those halves. Now we're going to start up here with the corpus callosum. And we will define this later, but the corpus callosum is essentially this thick band of fibers that connects the two hemispheres. We have about 200 million axons of sensory neurons and motor neurons that cross sides at this place. The next one down in the middle area there is the thalamus. Continuing on around, we have the cerebellum. Moving over down to the bottom on our left-hand side here, this is actually where it becomes the spinal cord. Now that you'll notice here, the nerve tissue is actually continuous as it moves from the spinal cord up into the brain area. We basically have the brain divided into three sections. We have the brain stem in this area, we have the cerebrum, and then we have the cerebellum. So the spinal cord is actually contiguous with the portion of the brain stem. There's no break in that neural material. The only difference is once it goes through this area here, this is actually a big hole in the skull. It's called the foramen magnum. The word foramen means hole and magnum means big, so literally the word means big hole. We actually consider it part of the brain at that point and not part of the spinal cord. So the first structure as part of the brain, after it passes through the foramen magnum, is the medulla oblongata. Above that is kind of the oval-shaped area called the pons. The next structure here is this little bulb on kind of that stalk. We're going to label it, but we won't talk about it until we get to the endocrine system, and that's your pituitary gland. It's actually basically a dangling piece of brain tissue to a certain extent. The pituitary gland is branched off of the hypothalamus. And then the final structure, it's pointing to a single section, but it actually represents kind of all of the ridged area here, and that is the cerebrum. Now the third diagram that you have, we actually want to take a look at the regions of just the cerebrum. So that's what we're looking at here. All right. And the nice thing about learning these is once you learn these, you're actually learning as well about the different bones, because the bones and the regions of the brain share names. So the first one is right behind your forehead, and that is the frontal lobe. Behind the frontal lobe is the parietal lobe. Kind of behind and below the parietal lobe is the occipital lobe. And finally, kind of at the sides, is your temporal lobe. Now these lobes are actually separated from each other by some key features. So the first feature we're going to look at separates the parietal lobe from the frontal lobe, and is this dark line right there. This is referred to as the central sulcus. So how is this different from a fissure? Well, a sulcus is a shallow depression. So the way I remember it is sulcus has an S and shallow has an S, whereas a fissure is deep. Now this central sulcus is not the only sulcus that we have. We also have sulci here. Okay, these little grooves, these are all sulci. Okay, but it's just this main one. You can see it extends for a lot longer than some of these smaller ones that we have. The next one is kind of off the back here. It's in this region. And this is the parietal occipital sulcus. The last one we want to look at is here, and this is the lateral sulcus. Now if you look at the cerebrum as a whole, we pointed out that it has some key sulcus, and also some smaller ones that I also drew on. But then you also have kind of this area in here that would be kind of like a raised area actually of tissue. It gives kind of that noodly or folded garden hose appearance to the cerebrum or the cerebral cortex. And so that raised area is called a gyrus. And that's any raised area on the cerebrum. Okay. So those are the three diagrams, and you would either have done in class or you can get off of my Moodle page the functions, because you have the table in your notes, and it should contain, or you should have gone through the activity where you did the different functions. So I will list them and kind of go through them quickly here, just kind of showing them on the screen, because this was actually a hands-on activity that you did. Okay. Now what I want to do is I actually want to point out a couple of groupings of structures that exist in the brain based on those structures. The first one is the brainstem, which I had mentioned earlier. The brainstem consists of the midbrain, the pons, and the medulla oblongata. So those three structures on your chart there would consist of the brainstem. So you may want to mark those somehow on your chart so that you don't forget that. The other one that you probably saw in the book is referred to as the diencephalon. The diencephalon is the epithalamus, the thalamus, and the hypothalamus. Keep in mind that that prefix epi means above, so the epithalamus is above the thalamus, and hypo means low or below, so it is below the thalamus. Now, some other things that have to do with the brain. The first one is the concept of hemispheric lateralization. Okay, this is basically where we specialize one hemisphere of the brain to perform a function that we don't perform on the other one. So this is actually more complicated than just simply my right side of my brain controls my left hand, because it works the same for the opposite side. These are actually functional things. Remember that the two hemispheres of the brain are connected by the corpus callosum, which it consists of about 200 million or more axons of both sensory and motor neurons, and we had pointed that out on the diagram. So if we're talking about hemispheric lateralization, what do we mean? Well, let me give you some examples. For instance, the left hemisphere of the brain controls writing if you are right-handed. If you are left-handed, obviously your writing center is going to be on the other side. So that's one that actually can switch just depending on your kind of hardwiring that you have. In addition, the left side has your language center. It also helps you perform mathematical calculations, logic, as well as speech. Now, if you look here, mathematical calculations and logic, a lot of times people who are good at math and science are referred to as left-brained, and that would be why. Those centers in those left-hand side of the brain tend to be very, very well developed for them. So what specialized areas do we have on the right-hand side of the brain? Well, if I am right-handed for my writing, then I do most of my touch analysis with my left. So the touch analysis side, if my writing center is on the left side, then I do most of my touch analysis on my right side. So I tend to use, for instance, I am right-handed, so I tend to use my left hand a lot to help me analyze things in terms of when I'm touching them. If I gave you a box where I said reach inside and figure out what that is without looking at it, you would actually use your left hand a lot more to get sort of the big picture of those features than you would your right. The right hand side also helps us to understand our three-dimensional relationships, our spatial relationships. This is one of the last centers of the brain to develop. And for some people, it barely develops. The last one is emotion interpretation. So certain people who have certain disorders, certain forms of autism, for instance, they cannot actually read people's emotions. That center in their brain is just not developed. So they have a very, very hard time with that. Now, in addition to hemispheric lateralization, with the brain we can talk about something called an electroencephalogram, or EEG. This provides a printed, or nowadays digital, record of the electrical activity of the brain. So you can actually monitor this, and what it does is when you print it out, it kind of looks like the wave pattern of action potentials. So that's the pattern that we look at are brain waves. They're literally the action potentials that are taking place. The types of brain waves are going to be tied to your level of consciousness as well as your developmental milestones that you have already achieved. We do use this as a diagnostic tool for certain brain disorders, and we do have 4 different types of waves. The first one are the alpha waves that are found in normal resting adults. We have beta waves, which indicate intense concentration and problem solving. Theta waves are typical in children or frustrated adults. When we think about it, a lot of times kids get frustrated because they want to be able to do something, especially younger kids, and they don't know how to ask. Their vocabulary hasn't developed enough to be able to communicate, so they get very frustrated. And you'll see the theta waves in them. Delta waves indicate deep sleep and certain pathological conditions. Remember, pathology indicates disease, so certain conditions will pop up delta waves when we're not supposed to see them. And these are what the different waves look like: the alpha waves, the beta waves, the theta waves, and the delta waves. And just to give you a sense of scale, for each of those, it's about 1 second. So the waves obviously happen very, very differently depending on the situation. Now, oftentimes why we do an EEG is because of something called a seizure. A seizure is a temporary cerebral disorder that can occur. There is a recurring seizure disorder that we have, and that is called epilepsy. What a seizure does is it changes the electrical activity of the brain that can actually be seen on the EEG. Now, how this happens, we're not quite sure always what triggers it, and that's part of what doctors a lot of times are trying to diagnose, is actually what triggers it for their patients. But it's almost like dropping a pebble in a puddle. It starts in one area, which would be where the pebble kind of plunks into the water, and it spreads outward from that epicenter in all directions like a wave across the cerebral cortex. So the signs and symptoms depend on the regions involved. You can have what's called a grand mal seizure, which involves convulsing and if you think of something like on a Hollywood drama with somebody having a seizure, that's very, very typical. But they can even be something as simple as a pause while their brain kind of resets. There are many different types of seizures. They don't all have to be this big grand mal type seizure. Now, seizures obviously are very disruptive to a lifestyle. So in severe epilepsy, where you're having several seizures a day, you can actually cut the corpus callosum to try to alleviate some of the symptoms of the seizures. Basically, what cutting it does is it prevents the seizure from jumping to the other hemisphere of the brain. There is some retraining and relearning of certain activities that the two sides coordinate with each other that has to happen, but it's not like a lobotomy where you're actually removing part of the brain. it's one of those things where the patient and the doctor have to work very, very closely with each other to decide what is best based on the current situation and level of impairment versus the side effects of the treatment. The last thing we want to kind of talk about with the brain, and specifically with the brain, is memory. Memory is basically stored bits of information that we hold on to. We can talk about memories being fact memories, which are very specific bits of information, like a phone number, social security number, or computer passwords. Those also tend to stay with you for a while too, but that's not what we're talking about. We're not talking about duration here at all. Versus skill memories. These are learned motor behaviors that can get incorporated eventually into an unconscious level. Things like writing, eventually it is a learned motor behavior that eventually you don't have to think about, you just do. Other things are like tying a shoe or playing an instrument. But how you're more familiar that we classify memory is based on its duration. So we can talk about short-term memory that includes small bits of information that don't last long. I want to order a pizza, so I look up the number to the pizza place in the phone book, and I only remember it as long as it takes to dial the phone. Once I'm done, I discard that. However, something that is in short-term memory can, by repetition, get moved into our long-term memory. So let's say that I call that pizza place every Friday for a year. Well, hopefully, probably somewhere along the line, I will remember the phone number and not have to look it up all the time. That bit of information has been placed into my long-term memory. Long-term memories last much longer, but they do fade with time. They're not as clear, you don't get immediate recall, and they can be a little bit fuzzy. They are often stored in association areas, and my favorite example of this is actually one of my personal ones. Every year at Christmas, one of the things I make is Chex Mix, and it really, the combination of the cereal and the butter sauce that I put on there with Worcestershire and all that kind of good stuff makes a certain aroma that my kids love. Well, one year for my daughter's birthday, I decided to make some Chex Mix. They were going to a corn maze, and so I made some Chex Mix to snack on at night because we were going to a flashlight maze. So my younger one, as I'm making this, comes downstairs, inhales very deeply, and goes, "It smells like Christmas." So that smell triggered the association area for that memory of Christmas. So that's what I'm talking about. Now, amnesia can be a loss of memory due to either a disease or some sort of trauma. The effects of amnesia depends on the regions that are affected. So it's usually not complete amnesia, it's usually partial amnesia, and some of it is recoverable just depending on the way that the memory was originally lost. If you damage your thalamic structures, so the epithalamus, the hypothalamus, and the thalamus, that will actually affect your storage of memories and can actually prevent you from storing new ones. And that can actually be a very confusing situation for the patients. The last thing that I have in this segment is this video. I'm going to pause it. This video clip will be available on the Moodle page so that you can review it. And that's going to kind of wrap up our in-depth look at the brain. We will be taking a look at this, the brain more hands-on when we dissect the sheep brain.